So in this module, as we have discussed in our previous module, that some ingredients are directly related to the growth of the microorganism involved in fermentation process, but some ingredients are required as precursors, inhibitors and inducers. In previous module, we have seen about and we discussed about the precursors. But in this module, we will talk about the inhibitors. So what are the inhibitors? As the name indicates, that those substances which inhibit the growth or the action of any enzymes during the fermentation process. So when we talk about the inhibitors added to the fermentation process, that is specific for some specific products. So the example, the metabolic intermediate which is normally metabolized is accumulated. So when any organism go normally, that automatically that will grow according to increase the biomass and normal metabolic pathways will carried out. So in case of some desired product, if we take the example of the production of citric acid, and we know that the citrate is the major metabolic step in citric acid cycle. So if we inhibit that enzyme which lead the conversion of citrate into the isocitrate, then if we inhibit that enzyme which help to pass on the citrate into isocitrate, then we can have the accumulation of the citric acid inside the cell. So then automatically, if that citrate or a citric acid which accumulate in the cell, then automatically that increase the production of citric acid. So the same way, there is so many other example of inhibitors that are mostly used for different products. You can see here in this slide that one of the earliest example for the microbial production of glycerol. So the glycerol production depend upon the modifying the ethanol fermentation. Normally we know that glucose is converted into ethanol fermentation. So it can be modified into the formation of glycerol just by removing the acetaldehyde or the inhibitor of that. So the addition of sodium bisulfite to the broth lead to the formation of acetaldehyde bisulfite. So the addition compound is sodium hydroxyethyl sulfite. So since the acetaldehyde is no longer available for reoxidation of NADH2, its place as an hydrogen acceptors, then dihydrogen acetone phosphate produced during glycolysis. So the product of this reaction is glycerol 3-phosphate, which ultimately converted into glycerol. So just by the addition of sodium bisulfite that inhibit the availability of acetaldehyde, which lead to the formation of ethyl alcohol. So if we remove or we inhibit that compound, that can lead to the production of our desired product. So just the inhibitors and precursors are the regulators. We can say that metabolic regulators for the fermentation process. Actually, the fermentation process is a chemically governed process using different microorganisms. Actually, we are controlling the metabolic pathways of that specific organisms which we are using during fermentation process. So the application of general and specific inhibitors are illustrated in the table. In the next slide you will see. So in most cases, the inhibitors are very effective in increasing the yield of the desired product. So number of studies have been made with the potential chlorination inhibitors such as bromide to minimize the chlorotetracycline production during the tetracycline fermentation. So there are a number of various examples we can quote, but in this slide you can see that different products and the inhibitors and their main effect, that how that inhibitor affect during the fermentation process. Here, on a first step, as we have already discussed, for the production of glycerol, and the inhibitor is sodium bisulfite, that the effect is acetaldehyde production repressed in case of Saccharomyces cerevisiae. In case of the tetracycline, bromide is the inhibitor, and you can see what is the function of that. In case of Streptomyces, so when we are dealing with the glutamic acid, for penicillin is the inhibitor, which is basically the deal with the wall permeability, and the organism is Micrococcus glutamicus. In case of the citric acid production, I have already given you the example of the inhibition of that enzyme. So the alkali metal, phosphate, and the pH below 2, that is basically inhibit, and that effect on by which the oxalic acid is repressed, and the organism is mostly used Aspergillus niger, that is filamentous fungi. In case of the valine, and there are so many inhibitors that affect with different inhibitors, and then you can see here the rifamycin B, then diethyl by so you can see here that diethyl barbiturate is an inhibitor. So that is mostly used, this Narcordia is the organism. So inhibitors are also used not only direct on effect on the enzymes, but in some cases they affect on the permeability of the cell wall or a cell membrane. So ultimately that lead to the excretion or enhance excretion of those product which we want to produce during fermentation process. So the role of the inhibitors are indirectly on the regulation pathway of the microorganism in order to achieve our desired product.